So my name is Megan, and I'm a PhD student at UIC, and I'll be presenting perfluoroalkyl substances in fish consumption in the Great Lakes Fish Consumer Study. So we'd just like to acknowledge the Wisconsin State Health Department, led by Dr. Anderson, for initiating this study, as well as our grant support. So PFAS are a very large group of man-made chemicals that do not occur naturally in the environment. There are many industrial uses and applications for PFAS due to their chemical properties. They're very stable chemicals with low volatility. The properties make them really attractive for industry use, but unfortunately bad for the environment. Their stability and long half-lives cause PFAS to persist in the environment, and subsequently they bioaccumulate up the food chain. And PFAS have been detected in every type of environmental media, and as well as globally, including the Arctic. So PFAS are frequently detected in serum samples identified in the National Health and Nutrition Examination Survey, or NHANES. NHANES is a representative of the US population and run by the CDC to assess the health status of both children and adults in the United States. Studies have shown trends of decreasing PFAS levels detected in US serum samples, as shown in the graph on the right. But these are mostly in PFOA and PFAS. And other studies have shown that other PFAS species are increasing in US serum samples. PFAS have a very long half-life in humans, similar to other persistent pollutants like PCBs. PFAS exposure has been linked to several adverse health effects, including cancer, endocrine disruption, and issues with metabolic function. Seafood consumption from contaminated waters may be a significant source of PFAS exposure. PFAS biomarkers have been associated with both consumption of finfish and shellfish, as seen in the figure to the right. It's the study conducted in my advisor's lab, assessing the proportional changes in the geometric means of PFAS in seafood consumers compared to those who do not consume seafood, using the NHANES data cycles from 2007 to 2014. And several dietary studies have also identified finfish consumption as the most significant pathway for food-related intake of PFAS in comparison to beef, poultry, dairy, etc. PFAS are ubiquitous in water, in the water and sediment and biota of the Great Lakes. One study from Reppel et al. Found that commercially purchased fish that were sourced from the Great Lakes had the highest level of PFAS. This is shown on the graph on the bottom left, where the orange bars depict the levels of PFAS in commercially purchased fish. Federal, state, local, and biomonitoring programs measure contaminants in the most common fish in the Great Lakes. Typically, the most common fish measured is lake trout, which is the top predator in every lake except for Erie, where walleye is the most common fish and is also the top predator. Summary data from Ramukal et al. Showing PFAS levels in fish from different Great Lakes identified that the contamination in lake trout was highest in Lake Erie in Ontario, while lowest in Lake Superior. About 4.2 million adults in the United States that border the Great Lakes consume Great Lakes fish, with around 500 of those that consuming 2 or more meals per month. 5 state health departments led by Dr. Anderson formed the Great Lakes Consortium in 1992 to assess critical pollutants in anglers. Participants were recruited from licensed charter boat captains, and the referent population was drawn from the same geographic areas as the charter boat captains, who had little to no Great Lake fish consumption. And there was another subgroup of Wisconsin anglers that only fished inland bodies of water, not the Great Lakes. And follow-up for exposure and health biomarkers was conducted through 2004. And Great Lakes fish consumption was shown to be a significant exposure source of both PCBs and DDE. Thus, our objective was to assess if serum PFAS levels are associated with self-reported fish meals in a cohort of frequent and infrequent Great Lakes sport fish consumers. This was a cross-sectional analysis of the Great Lake fish consumer study cohort, frequent and infrequent Great Lakes sport fish consumers, using the blood samples that were collected in 2004. There were 474 stored blood serum samples, and they were tested for 12 PFAS species by the CDC. The detected methods were described elsewhere. Observations below the limit of detection were imputed as the limit of detection divided by the square root of 2. We removed 6 observations due to missing covariates. Participants completed detailed questionnaires on their health history and fish consumption, including fish meals consumed in the past year by fish species and source. So the PFAS compounds were log-based 2 transformed for analysis to help normalize the distribution. And the predictors were Great Lakes sport fish meals, commercially purchased fish meals, and sport fish meals from other inland bodies of water. And our covariates were age, sex, BMI, and education level. So the median age in our sample was 58 with a median BMI of 29. And there were significantly more males than females in the analysis. And we did actually end up finding differences in male and female total Great Lake fish meal consumption, but there were no differences observed in total commercially purchased fish meals, nor the inland fish meals. So the geometric mean for total sport fish meals from the Great Lakes was about 6. And the geometric mean was about double that for the total commercially purchased fish. The median of the other inland bodies of water was 0. So there wasn't much consumption, as much fish consumption as there was from the Great Lakes and commercial fish. And so these are the geometric means for each PFAS series in the serum concentrations. N-PFAS was the highest serum detected species, followed by SM-PFAS and N-PFOA. This is consistent with what's reported in the literature, and N-PFAS being the species that's often reported with as found with the highest concentrations. And all serum samples for SP-PFOA were under the limit of detection. So this is the graph showing the percent serum PFAS levels for increases in 5 sport fish meals per year, and with the highest increases seen in PFDA and PFAS and PFUNDA. Nearly all of the PFAS species showed higher percent increases in the Great Lakes sport fish meals consumed compared to the commercially purchased sport fish meals. And here we show the percent increases in serum PFAS levels in 1 and 2 or more meals per week compared to 0 meals per week. We didn't see any significant increases for 3 of the PFAS species at the leftmost on the graph. And the highest percent increases were seen in Great Lakes fish meals per week compared to total inland fish meals and sport caught fish meals. And again, the highest increases were seen in PFDA and PFAS and PFUNDA. There are some limitations with our study. This was a cross-sectional analysis, and the data was collected in 2004, so the PFAS levels may have changed since then. Recall bias could be present as fish consumption was self-reported on questionnaires, and there might be residual confounding from other unmeasured exposures. So in conclusion, we found significant increases in serum concentration of several PFAS, and they were seen for both Great Lakes sport caught fish and commercially purchased fish meals, with larger increases seen in the Great Lakes fish. So where we're going next with this is to examine specific consumption by fish species and Great Lake, where the fish came from. And we also want to assess the correlations of the PFAS biomarkers with other measured contaminant biomarkers in the study sample, which include PCBs, PBDEs, and DD and mercury. Thank you.
Great, thanks. We have about 4.5 minutes for questions.
Okay, the first question we have is, why use BMI when it does not well correlate to body fat?
We wanted to control for it because we did see some differences in, especially when we did see differences in between Great Lake fish consumption and commercially purchased fish consumption when we looked at BMI.
Next question is, it is pretty interesting since it is related to the Great Lakes. How can we combat PFAS in aquatic environment?
I'm not really a remediation specialist, but so I'm not really sure how to answer that. I'm sorry.
Were you able to notice if there were any differences in PFOS between species in the same environments?
So we haven't looked at Great Lakes from where the fish came from yet. So I'm not sure. We're not sure yet.
So being an outreach specialist person, I was curious what kind of materials the cohort was given before and after your study.
So they were given detailed information on fish advisories for all of the different lakes. They were also given pamphlets about healthy fish to consume versus fish that might have more contaminants, but really specific to PCBs and PBDs.